In this video, we're going to talk about inheritance, and in the first section of inheritance, we need to recall and remember a whole lot of definitions and understand some terminology. So to start with, we're going to go back a bit and talk about, firstly, proteins. Remembering that proteins are biological molecules. They are polymers or large molecules made from monomers called amino acids. And proteins have many uses within the body, including enzymes and antibodies. And at all of these applications, there's a whole number of them listed here. We know that what is important with proteins is their shape. The first stage in determining the shape of a protein is the amino acid order, or the sequence of amino acids in the chain of the polypeptide. And we can see that in the diagram. The second type of biological molecule we're going to talk about is DNA, or deoxyribonucleic acid. This is the biological molecule that's responsible for inheritance. And small sections of the DNA are called genes. We talk about genes and genetic inheritance. And there we remember the structure, the double helix of DNA with the phosphate group and the sugars in between, and they're joined to bases. Remembering that those are called nucleotides. They join up in a long strand of DNA. So inheritance, the definition is the transmission of genetic information from one generation to the next generation. A chromosome is just a thread of DNA made up of a string of genes. So chromosomes are how DNA is actually found in the nucleus of the cell, all bunched up into sections as we see in the diagram here. When DNA is stretched out, the different sections of DNA are called genes, and one gene will code for or will make one protein. So a gene is a length of DNA that is the unit of heredity. It codes for a specific protein. And these genes will be passed on or may be passed on from one generation to the next. There we see the cell with its nucleus, the chromosomes, which when they're stretched out are made of DNA, and a portion of that DNA is called the gene. The gene, we're going to learn about how the gene makes a protein. And the last term that you need to learn, and is probably very new to you, is the word allele. An allele is two or more alternatives of a form of gene. So, for example, the gene for eye color, there are many different alternatives. Two alleles of that gene would be brown eye color or green eye color. Just to explain a few more terms and define some other words. In DNA, we remember that there are 4 bases, A, T, C, and G. We're not going to worry you with the names of them. And that the bases in DNA pair up with their complementary base. So A always pairs with T and C with G. A with T, or sometimes A with U, and C with G. The sequence of bases on the gene is important, and mRNA is going to be made from DNA. We're going to talk about that now in protein synthesis. So there's our DNA, double helix. The mRNA copies information from the DNA and carries it outside the nucleus, where the ribosome uses the mRNA to build protein from amino acids. So DNA is then turned into a section of mRNA. Okay, what you need to know is these 3 bases next to each other, the first 3 being guanine, uracil, and adenine, GUA, is called a codon. Every codon, every 3 bases next to each other, codes for one amino acid. A codon codes for a specific amino acid. And once they do, those amino acids will be joined in. Proteins are made by ribosomes and RNA, and protein synthesis takes place in the cytoplasm. So protein synthesis, in a nutshell, we'll discuss it in more detail in a short while. DNA is transcribed into mRNA or messenger RNA. This process is called transcription. And then mRNA, this strand, this template that has come from the genes in your cells, is used to make a protein, and that process is called translation. So these three things are linked. DNA transcribed to our mRNA, which is then translated into a protein, which folds up and is useful within a cell. In this section of the video, we're going to talk about protein synthesis, and we're going to discuss the two main stages of protein synthesis, transcription and translation. So firstly, we'll deal with transcription. Transcription occurs inside the nucleus of the cell, and basically it's the process of copying, making a copy of DNA and converting it into RNA. And in particular, this strand of RNA is called mRNA, or messenger RNA. So let's look at the details. If we look at a strand of DNA, it's a double strand. What happens when we're about to make mRNA is this strand, a certain section of it, called a gene, unzips. And when this happens, an enzyme called RNA polymerase is able to bring in nucleotides of RNA and join them up with the complementary bases on the DNA. So as you see here, the adenine joins to the thymine, and so on. As they join in, they form a strand of RNA, which is called mRNA. Basically, the DNA gene is copied into mRNA. Once transcription is complete, the mRNA can leave the nucleus, and it moves from the nucleus through the nuclear membrane into the cytoplasm. In the cytoplasm, this mRNA joins up with a ribosome, and once it joins up with the ribosome, small strands of tRNA, a different type of RNA, can bring in the amino acids and start to build up, using the template of the mRNA, start to build up the protein that is to be made by this gene. So we just build these all up into one long strand. To find the ones that fit. One long strand of amino acids. Oh, not that one, leucine. Then this one. And when the last amino acid is added in, it stops making the protein, and the protein is released and folds into a specific shape. We always talk about the shape of proteins and how important that shape is. The primary structure of protein is the order of amino acids. Once it folds into a specific shape, it is able to do a specific job, maybe the job of an enzyme or an antibody or a plasma protein or a protein that is involved in active transport in cell membranes. So that is the process of protein synthesis.